All right, this lecture is on hearing and equilibrium, functions that are governed by your ears and the inner structures surrounding the ears. If we look at the general functions, we find that the ears are responsible for a sense of balance, that is equilibrium, and there's two ways that you're keeping track of equilibrium. Static, that is while you're standing still, or in other words, basically a reference with respect to gravity. So which way is up? And the dynamic equilibrium is your balance with respect to spin or motion. So balance is one set of functions, and then the other set of functions obviously is hearing for the ears, so your sense of hearing. So here we see the ear, and it is cone-shaped, funneling sound into the external acoustic meatus. So this cartilage structure that is covered by skin is sometimes referred to as a helix. Sometimes it is referred to as an auricle. So it takes on a couple of different names. We also see some other areas here, and that is the tragus, that little point that's right there, and then the antitragus, and then obviously your lobe or lobule. But the main focus here should be that the ear, the cartilage covered with skin, is a cone shape to send sound waves down the external acoustic meatus. So let's get a little better look at this. Here we can see the pinna or auricle or helix is described here. Again, the conical shape to get sound waves to travel down the external acoustic meatus. Lining the external acoustic meatus, there will be a number of ceruminous glands, and they will produce a modified sebum or oil or wax, and that is earwax. So ceruminous glands line the external acoustic meatus. There's also hairs that line that area, and the hairs with the wax on the hairs prevents objects, bacteria, other foreign materials from getting down deep into the ear where they can come in contact with the delicate tympanic membrane. So the ear is divided into three different areas. There is the external ear, and that is everything from the tympanic membrane out. Then there is a middle ear, which is just on the other side of the tympanic membrane, all the way until we get to the oval window and the round window. So this space in the middle is the middle ear. And then all the way on the inside, we have structures of the inner ear. So the inner ear is going to have three different areas. It'll have the cochlea, which will be involved in hearing, and then it will have this broad area across the middle, which is referred to as the vestibule, and then it'll have these coiled tubes over here, the semicircular canals. The vestibule and the semicircular canals are involved in balance, whereas the cochlea is involved in hearing. The external ear brings us from the outer ear, the auricle, all the way down the external acoustic meatus, all the way up to, but not including, the tympanic membrane. The tympanic membrane is going to be the division between the external ear and the middle ear. The tympanic membrane is a delicate membrane. It's a living tissue, and it will bounce and vibrate based on the sound waves that are entering through the external acoustic meatus. Those sound waves are then transferred to the middle ear, and in the middle ear we see some bones, some auditory ossicles. We see the malleus, the incus, and the stapes. So the malleus, the incus, and the stapes are three bones, and they are attached to each other by synovial joints. So this synovial joint right here between the incus and the stapes is just about the smallest synovial joint in your entire body. A synovial joint is a fluid-filled joint, and that allows these bones to rock on each other and not wear down. They're rocking on each other because the tympanic membrane vibrates, which rocks the malleus into the incus and into the stapes, and then the stapes bounces over the oval window, and then the oval window and the round window are the division between the middle ear and the inner ear. And what we see is if the oval window is vibrating, it causes that vibrational energy to enter into the inner ear and eventually work its way down into the cochlea, where we will turn those sound waves and sound energy into an action potential that will travel down the cochlear nerve. Now realize that the cochlear nerve is not one of your cranial nerves, but there is one that you should recognize, and that's vestibulocochlear. And we have the vestibular nerve coming from the vestibule and the semicircular canals, and that's governing, again, balance. And the cochlear nerve coming from the cochlea for hearing, and those are going to combine together to form one of your cranial nerves, the vestibulocochlear nerve. You also see running right along through here is also the facial nerve, but that's not, see, it's cut off, it's coming from somewhere else on the face surface. So let's follow a sound wave in through the ear, moving the tympanic membrane, rocking the malleus, the incus, the stapes, vibrating the oval window, and then we start to vibrate the fluids inside of the cochlea. But before we follow that any further, let's go back to the middle ear and finish that up. So within the middle ear, we have an air-filled chamber, and that air-filled chamber is attached to this tube referred to as the auditory tube or eustachian tube. The eustachian tube is connected to the throat and will allow for an equalization of the pressure on the within the middle ear at the same time as the external ear. So the external ear and the middle ear, we want to have the same pressure so that when the tympanic membrane vibrates, it's not bowed out or bowed in based on a pressure difference. So the eustachian tube equalizes pressure. So when you chew, those muscles of mastication or chewing are running right along the eustachian tubes and squeezing it and causing air bubbles to move up or down. When an air bubble enters into the middle ear, it pops, and that would cause a little vibration within the tympanic membrane, and you would hear a pop. So your ears popping are a regulation of pressure between the middle ear and the external ear and making sure they are the same. Now let's continue on with hearing and go to the inner ear and see what's happening in the cochlea. So here we see the oval window, and as it would vibrate this fluid that is inside of the cochlea, and the fluid is the scala vestibuli fluid, and that scala vestibuli fluid would vibrate, and as that scala vestibuli fluid would vibrate, it would cause this vestibular membrane to vibrate. We got this blown up over here, so here is that fluid within the scala vestibuli, and then here is the vestibular membrane. So the vestibular membrane is going to vibrate, causing the fluid within the cochlear duct to vibrate. The cochlear duct leads all the way down to this basement membrane called the basilar membrane. The basilar membrane has sitting on top of it a set of cells that are supporting cells, and then these very specialized neurons called hair cells. They are actual neurons, and these ends are modified dendrites into these cilia, or in this case stereocilia, and those modified dendrites look like little hairs projecting up. So the cells are neurons, but they are referred to as hair cells. So going back to the vibrations, the cochlear duct fluid was vibrating, which caused the basilar membrane to vibrate. And so this spiral organ, which is made up of the supporting cells and the hair cells, the spiral organ starts to bounce up and down. And as it bounces up and down, these little cilia, the hair-like structures, smash into the tectorial membrane. So the tectorial membrane is a stationary membrane right above the spiral organ. The spiral organ starts to bounce and smashes the stereocilia into the tectorial membrane, bending those hair cells, sending an action potential through the cochlear nerve, and goes to your brain saying, you heard something. Now, if we look at the basilar membrane, we see that the basilar membrane has differential elasticity. The differential elasticity that we see with the basilar membrane is such that near the oval window it is very stiff. So the basilar membrane is very stiff near the oval window, and as you go out further and further towards this point of the cochlea called the helicotrema, further and further out towards the helicotrema, it becomes more and more flexible. So very high frequency sounds can cause the stiff basilar membrane to vibrate, and then the sound wave travels in there. But if it is a lower tone or a lower frequency, it keeps moving until it can cause the basilar membrane to vibrate. So the differential elasticity of the basilar membrane means that you hear high frequency sounds or your highest frequency sounds near the oval window, and then lower and lower and lower sounds until you get all the way out to the lowest sounds, which are picked up at the helicotrema. You can see that there are some sound waves that are traveling on this other side, and that is the other side. You'll have those sound waves travel until they exit out the round window. The round window is then just a relief valve, allowing out that vibrational energy. You wouldn't want those sound waves or that vibrational energy coming back this way and towards the oval window. We just keep moving all in one direction and vibrate out here and get the sound waves gone, vibrational energy gone through that round window. So here we can see, because of that differential elasticity, that the highest frequencies at 20,000 hertz is being picked up near the oval window, and the lower and lower frequency sounds down to 600 and below 200 hertz are really at that helicotrema, that point within the cochlea. Next, we move over to equilibrium. For equilibrium, we are concerned with static equilibrium, and that is equilibrium while you're standing still and keeping track of the ground, or which way is up relative to gravity. So we have a system of the vestibule, which is within the static labyrinth, and within the vestibule there's two distinct areas. There is the utricle, and then there is the saccule. And the utricle is parallel to the base of the brain. And so when you are standing up, the utricle is keeping track of which way is up relative to gravity. And so if you could imagine a table laid out in front of you, and then you could throw Jell-O all over the table. And if you lifted up the table on the right side, it would lean to the left, and the Jell-O would slide to the left, and that would say that you were leaning to the left. If you leaned it the opposite way, you'd be leaning to the right. And so that's basically what's happening within that utricle. The saccule is perpendicular to the base of the brain. So when you lie down, then the Jell-O within that area of the saccule would be spread out evenly, and then you would know whether your feet were getting higher than your head, or whether your head was getting higher than your feet, or whether you were rolling out of bed to the right or rolling out of bed to the left. So the vestibule, we'll take a closer look at that and the specialized areas of the utricle and the saccule in a moment. Those are for static equilibrium, again, structures within the static labyrinth. Within the kinetic labyrinth, that tissue contains the semicircular canals. And the semicircular canals are 3 sets of tubes that are oriented at right angles to each other. So if you had one line running this direction, and then right angle to that would be this direction, and then a right angle to both of these would be if you had something running right through the screen on the computer. So the third dimension is up, down, and then straight through the middle. Right and left, and then straight through the middle. So 3 sets of tubes oriented at right angles to each other. So this is going to help us to detect spin in 3 dimensions. So if you were doing a somersault, that would be 1 dimension. If you were doing a cartwheel, that would be another dimension. And then spinning like a ballerina would be a 3rd dimension. So when we look at the utricle and then we look at the saccule, again, the utricle is parallel to the base of the brain, and the saccule is perpendicular to the base of the brain. Okay, that's within the vestibule. Within each of those, the utricle and the saccule, there is a specialized area referred to as a macula. So there is a utricular macula and a saccular macula. And so here was that idea of the jello laid out above on top of a table. So our table here is interesting because for the table what we see is a number of supporting cells within this utricular macula and then hair cells embedded within this table-like structure of cells with the gelatinous matrix spilled out over the top. So if you lean this table to the right or to the left, that causes a bending of your hair cells again, sending the signal, you're leaning to the left, you're leaning to the right, you're leaning forward, you're leaning backward. So any dimension that you lean, right, left, forward, backwards, you're going to tilt the table, which tilts the gelatin, which bends the hair cells, and sends a signal that you are leaning in that direction. The saccular macula would become flat like this if the individual was to lay down. And as we suggested before, you would know when your head was higher than your feet, when your feet were higher than your head, when you were rolling out of the bed to the right, rolling out of the bed to the left. And again, the saccular macula would look exactly like this: hair cells, supporting cells, making a table with a gelatinous matrix on top. So here it is in practice. We see the utricular macula here, because this is his head, and parallel to the base of the brain. So we have the utricular macula laid out with the gelatinous matrix, and if that individual looked down, then that gelatin would slide down, saying you're looking down, you're looking down, you're looking down. So here it is in practice. The next area is the semicircular canals and keeping track of spin. So if we look at the semicircular canals, we see that we have the 3 tubes all at right angles to each other. They're fluid-filled tubes, and at each one of the ends of the tubes there's a little bulb, and the bulb is referred to as an ampulla. Here the plural, ampullae, referring to more than one of them. Within each ampulla, if we zoom in here, we see supporting cells surrounding a number of hair cells. So here we have our cilia sticking out the top again, the modified dendrite that is the hair cell, the modified neuron that is the hair cell. The bending of that hair cell triggers an action potential that goes through the vestibular nerves and says that you are spinning. So on top of the crista ampullaris, which is the area where we have the supporting cells and the hair cells within the ampulla, there is a gelatinous matrix here too. It forms like a flame shape, and so that's referred to as a cupula. And the cupula will, if water rushes in here, then it will bend the cupula, which will bend the hair cells and again send that signal. So if you are spinning, the fluid would move within the tube. So as soon as you turn, the fluid stays still, the tube moves, and you would bend the cupula as the water rushed into that area. If you were doing a somersault or cartwheel or spinning like a ballerina, you'd be getting different angles of spin, and therefore different fluid would be moving in different tubes, saying that you were doing a somersault, a backflip, spinning like a ballerina to the right, spinning like a ballerina to the left, or doing a cartwheel to the right or the left. So now think about this in terms of if you were spinning like a ballerina, and then the fluid initially when you start to move would go into one of these areas, depending on whether you were spinning right or left, and then eventually the tube keeps spinning and spinning and spinning, and you get to the point where the fluid catches up with the tube. So the tube and the fluid are now moving in unison. And then what would happen if the individual stopped? When the individual stops, the fluid keeps moving. And as the fluid keeps moving, it's sending a signal that says you're turning to the left, you're turning to the left, you're turning to the left. But as you try to walk, your eyes are telling you something different, and you stagger and you're dizzy and clumsily marching along. And so the sense of dizziness or disorientation with being dizzy is because the fluid is still moving, but you've stopped the tube. So here we have a gymnast, and she is flipping head over heels here, and that's causing the fluid to enter in, bend the cupula, sending the action potential through the bending of the hair cells and down into the vestibular nerve, which will join up with the cochlear nerve to give us the vestibulocochlear nerve. Finally, I just want to talk briefly about cochlear implants. In the medical field, we see some nice advances here where we can have an electrode that is allowed to pass through the tympanic membrane, through the the round window, and then wrapped up to the inside of the cochlea. And as that wire is wrapped up inside of the cochlea, it is able to stimulate those nerves along those areas. And so if you have an antenna and a transmitter and a receiver out here inside, just underneath the skin of the head, if you pick up sound waves there, high frequency or low frequency sound waves, if they are higher frequency sounds, you want them transmitted up in this region. And if they are lower and lower frequency sounds, you would want them transmitted or to stimulate neurons in this region. And so we can differentiate by how far down in that wire the contacts are located, and the contacts would interact with the neurons, the hair cells, and trigger them to fire based on what was picked up by the transmitter, receiver, and antenna within the cochlear implant device. So just showing you that there are some really cool advances that are happening, and that there are ways to think about this in terms of this is eventually, when we get into these neurons, it's an electrical system. So to bypass this mechanical system with some electrical activity is a very interesting solution.